have you on Planetterrian Daily, episode 111. It's July 5th, 2026. Let's get into today's research and health developments. Researchers mapped a brain circuit that couples deep sleep to growth hormone release and metabolic regulation. Researchers identified the neurofeedback loop that connects deep sleep stages with growth hormone secretion. The circuit explains how disrupted sleep impairs muscle repair, fat metabolism, and brain maintenance. Poor sleep interferes with this loop and contributes to metabolic and neurodegenerative conditions. The work points toward targeted interventions for sleep disorders tied to Alzheimer's and Parkinson's. Next steps include testing whether modulating this circuit can restore hormone rhythms in clinical populations. Source material comes from Science Daily. The newly mapped deep sleep circuit ties growth hormone pulses directly to the same neurons that regulate slow-wave activity. This feedback loop supplies a concrete mechanism for why fragmented sleep disrupts both muscle maintenance and cognitive clearance pathways already under study in aging biology. Because the circuit operates bidirectionally, interventions that stabilize deep sleep could simultaneously improve metabolic and neural outcomes. The finding moves the field from correlation toward testable targets for restoring hormone rhythms. Most people picture sleep as simple downtime that lets the body rest. In reality, deep sleep actively drives hormone release and tissue maintenance while you lie still. Right now, as slow-wave activity builds in the brain, specialized circuits trigger pulses of growth hormone that reach muscle, fat, and bone. A single night of restricted deep sleep can cut growth hormone output by roughly half compared with uninterrupted nights. That drop directly slows muscle protein synthesis and alters fat metabolism the following day. The practical takeaway is to protect the first half of the night when slow-wave sleep peaks by keeping the bedroom cool and dark and avoiding late caffeine. Future reports on sleep stage pharmacology will show whether drugs that lengthen deep sleep can safely restore these hormone rhythms without side effects. Now, shifting to a very different area of research. A study of people who reached 100 with preserved health identified a chemical signature in blood that differs from typical aging trajectories. The pattern involves specific bile acids and steroids associated with extended survival. This fingerprint adds a measurable marker to ongoing aging biology research. It fits within the tracked questions on reliable biomarkers that predict healthy longevity rather than chronological age. Follow-up work will test whether these metabolites can be tracked prospectively in larger cohorts. On a different note, disturbances in olfactory function frequently precede or accompany neurodegenerative processes. The connection arises because olfactory pathways intersect with regions vulnerable to protein accumulation and inflammation. Clinical data show that smell impairment can appear years before measurable cognitive decline. Researchers are examining whether olfactory testing can serve as an early, non-invasive indicator. Longitudinal cohorts will determine how strongly smell scores predict later brain outcomes. Source material comes from Ars Technica. Next up, University of California, Los Angeles researchers found that small cell cancers missing the RB gene become critically reliant on the transcription factor E2F3. Blocking E2F3 halted tumor expansion in laboratory models. Existing approved drugs may already target this dependency. The discovery identifies a vulnerability in aggressive cancers that have resisted prior therapies. Clinical translation will require confirming that E2F3 inhibition remains selective in patients. From genetic dependencies, we turn to an engineered tool. ETH Zurich researchers engineered a molecular switch activated by light to destroy receptors that let cancer cells enter a stress-induced dormant state. In laboratory lung cancer cells, the approach awakened the sleeping cells and rendered them vulnerable again. The method selectively acts on the receptors while sparing healthy tissue. It addresses a survival strategy used by tumor cells to evade standard treatments. Further development will examine delivery methods that confine the light activation to tumor sites. Shifting to basic cell biology, Heinrich Heine University Dusseldorf and LMU Munich researchers analyzed how bacteria insert proteins into cell membranes. Their findings reveal more biochemical overlap with eukaryotic processes than earlier models assumed. The work clarifies the conserved steps that move proteins across lipid bilayers. It refines understanding of membrane biogenesis across domains of life. Future experiments will compare the efficiency of these shared components in different organisms. Source material comes from phys.org. Finally, a much older biological record surfaces from ancient teeth. Analysis of dental remains from Siberia revealed the earliest known traces of Yersinia pestis. The find moves the documented presence of the plague bacterium roughly 200 years earlier than previous records. It shows the pathogen circulated in human populations well before recorded historical outbreaks. Genomic reconstruction from the teeth provides a baseline for tracking later strain evolution. Additional ancient samples will help map the geographic spread of early lineages. A German-Japanese team demonstrated optical writing of magnetic information in antiferromagnets using only laser pulses. The method eliminates the need for electric currents or external magnetic fields. It opens routes to energy-efficient data storage and processing devices. The breakthrough shows that antiferromagnetic order can be controlled at ultra-fast timescales. Next measurements will assess endurance and scalability of the laser-written states. Paleontologists identified a previously overlooked dinosaur specimen collected from Antarctica and stored for decades. The fossil adds to the sparse record of polar dinosaurs. Its preservation offers new anatomical details from a high-latitude environment. The discovery highlights how existing collections can still yield significant finds. Further preparation and imaging will clarify its taxonomic placement. Researchers isolated a bacterial protein whose calcium affinity changes sharply with small shifts in proton concentration. The finding illustrates a general rule for ion coordination that operates across pH ranges encountered in cells. It demonstrates how local acidity can switch protein function without altering the amino acid sequence. The mechanism helps explain calcium signaling under varying physiological conditions. Structural studies will test whether similar switches exist in eukaryotic homologs. Researchers trained convolutional networks on more than 120,000 photographs of salmon lice larvae. The models identified the parasites faster and with higher accuracy than trained biologists. The system addresses a major bottleneck in aquaculture health monitoring. It reduces the time needed to scan seawater samples for early infestation. Deployment trials will assess performance under variable field lighting and water clarity. American herring gulls retain mottled brown feathers for several years before acquiring adult plumage. Field observations show that this delayed maturation reduces aggressive responses from breeding adults. The plumage acts as a visual cue that the bearer poses no competitive threat. The pattern supports survival during the extended period before first breeding. Comparative studies across gull species will test whether similar signals evolved repeatedly. Astronomers detected a pair of Jupiter-sized worlds with densities lower than any previously measured. The planets belong to the superpuff class, yet exceed prior examples in size and fluffiness. Their existence challenges standard models of gas giant formation and atmospheric retention. Transit and radial velocity data constrain possible compositions dominated by voluminous hydrogen-helium envelopes. Additional atmospheric observations will clarify how such low-density structures persist. Physicists built a device that uses shaped laser fields to rotate molecules suspended in a superfluid. The technique allows controlled study of rotational dynamics at the quantum limit. It reveals how superfluidity breaks down when molecular motion exceeds a critical threshold. The method provides a new probe for frictionless flow at the atomic scale. Planned upgrades will extend the range of molecules that can be spun and measured. Projections of 1.7 million satellites in low Earth orbit will create persistent trails across telescope images. The cumulative light pollution and radio interference exceed thresholds that current mitigation strategies can address. The study quantifies expected losses in survey efficiency and transient detection. It calls for revised orbital policies that account for astronomical infrastructure. International coordination on brightness limits and spectrum allocation remains the next required step. New findings on sleep circuitry and blood metabolites continue to sharpen the map of healthy aging. Keep an eye on follow-up trials that test whether stabilizing deep sleep circuits can restore growth hormone rhythms in older adults. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Models and Agents for Beginners. Artificial Intelligence Explained Simply for Beginners and Curious Teens. Find every show free at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.